yEvo is a yeast evolution lab for the high school classroom. We perform evolution experiments by growing yeast and transferring them to higher concentrations of our chosen selection, like a drug or different nutrient source, to select for yeasts that are better adapted to that condition. This adaptation is due to random mutations that arose as the yeast grew. So in order to have more mutations for selection, we need to grow the yeast for several weeks. We do this by transferring a small amount of yeast from one tube into a new tube to grow more, at least once every week. At the end, we show that the evolved yeast grow better than the starting ancestral yeast, and then sequence the evolved yeast to determine what changes in their DNA may have contributed to their adaptation. Here is how to complete a round of transfers in your classroom lab. We will make observations about our culture, collect supplies, prepare our work area, carry out the transfer, and put our new cultures of yeast to grow for another week. First, we collect our tube of yeast that we grew previously. If the tubes have been sitting, the yeast will have settled to the bottom as a pellet. If, after sitting for an hour, the liquid is still cloudy, it is contaminated with bacteria. We know this because some bacteria can swim, but yeast cannot, so the yeast sink to form a pellet. We make sure to record everything we are doing, as well as any changes to the protocol or important observations, like if our yeast has not grown very much or if our culture is contaminated with bacteria. This will help us to figure out what we did in the future when it's time to analyze our experiment. Now we need to collect supplies. We have our previous culture. We also need a sterile cotton swab and a tube with media for our new culture. If your yeast grew well, you can select media that has a higher concentration of our chosen selection. Make a label with your group name, the date, and the media you use so that you can find your correct tube next week. To prepare for our transfer, we always wash our hands to keep our yeast cultures free from other microbes in the environment. We also light our Bunsen burner to create an area of sterile air so that no other microbes in the environment fall into our tube and contaminate it. It is okay if your classroom does not have Bunsen burners, but they help reduce contamination. Make sure to secure any loose clothing or long hair when working with an open flame, and then work within a 12-inch radius of the burner when your tubes are open. We are now ready to transfer our yeast culture. We take the tube of yeast that we grew previously and mix the yeast into the liquid by gently flicking the tube. Now we are going to use a sterile cotton swab to transfer yeast from our older tube of grown yeast into the new tube that we have prepared. Have a lab partner open the tube of yeast you grew near the Bunsen burner while you open a sterile swab. Then swirl the swab in the liquid to pick up the yeast, being careful to not touch the tube rim with your fingers. Then have your lab partner uncap the tube for your new culture and put the swab with the yeast into that tube so the tip is in the media, again being careful not to touch the tube rim. Swirl the swab a few times, remove it, and have your partner recap the tube. Dispose of your swab in a biohazard collection container. Place your yeast in a rack or roller drum so it will begin to grow again. It will grow at room temperature or in a 30°C incubator. After a few days, when you can see yeast growth in your new tube, repeat this process. More transfers gives more opportunities for helpful mutations to arise as the yeast grow, and will allow us to transfer into higher concentrations of the selection as the yeast evolve more resistance.